And if we could start making our way back to our seats in preparation for our next round of talks. All right, and it is now time for us to start the next talk. So please take your seats. All right. And our next presenter is Genevieve Anderegg on Morphos and Birdwings.
All right. Hello, everyone. I'm Genevieve Anderegg. Please call me Jenna. I am the Assistant Collections Manager of Invertebrates at the Denver Museum of Nature and Science. So I take care of our arachnology, marine invertebrate, and entomology collections. And this is a presentation about the Treves collection that our museum received recently. So Jean-Francois Treves is an amazing entomologist. He was born in 1930 in Brussels, Belgium, and he is still kicking. He was a mathematician of partial differential equations. And his academic advisor in university was Laurent Schwartz, who was also a really large lepidopterist, and donated his collection to the Paris collection or Paris Museum. And these two were best friends after Laurent was Treves's advisor, and they both were really into morphos and birdwing butterflies, basically the most beautiful megaleps in the world for collecting. So Treves taught at Rutgers and then retired and moved to Colorado with his wife, Ursula. And basically this donation was proposed to our museum in 2022, and we knew that it contained a lot of material that's protected under national and international treaties, and that we needed to assess it for legality in order to accept it into our institution, because this includes material that Treves not only collected himself, but also a lot of things that he bought through the insect trade. So the overall donation basically was loaned to us for a year in 2022, and it is a worldwide collection from the 20th century. And we knew we needed to assess it to make sure that it was collected in adherence to relevant national and international legislation, including CITES. I can never remember what that acronym means, so it's there on the screen for you. And a date to remember is that went into effect on the 1st of July in 1975. And this donation comprised 122 jars, 45 Schmidt boxes, and one giant metal trunk full of papered material for a total of over 5,000 specimens. So these were all loaned to our institution for evaluation. So I started this job in June 2022, and within the first couple of weeks, we headed on up to Golden, up the road into the mountains, to Treves's beautiful house in the mountainside there, and had to back this U-Haul down a slope into their backyard and then carry all of these drawers into the insect cabinets in the back of this truck to move them to our museum. They were all put on a rotating freezer schedule of 2 weeks in, and that was a lot of negotiation with our small freezer as well. And then immediately it was passed to me as my job to evaluate this whole donation to see if we could accept it. Because again, we knew a lot of these specimens were listed on CITES. So for the pin and papered specimens, I have 2 separate workflows. For the pin specimens, I knew what the CITES listed species were and what they looked like. And so it was much easier to identify those first and then look at their collecting information on the tags underneath to see if those collecting dates lined up with the legalities for collecting. So I inventoried all the specimens of listed species and compared the collecting data. And then for specimens of listed species that did not have collecting data, we actually had all of Treves's personal archive loaned to us, which was like a giant stack of letters that him and Laurent Schwartz and all of these people that he communicated with over the years, many, many decades, about their love of morphos and birdwings. And so one of my coworkers, Tiffany Nestle, who speaks French, actually translated these documents for me and looked basically for any references to specific specimens, and because of the dates on the letters, to see if that would lend us any more information on when Treves owned these specimens and if that was before or after their sighting date. And so we ended up inventorying 284 of about the 2,300 pinned specimens in the collection. And this, include, again, a lot of these really beautiful things like Troides and Trogonoptera and Morpho and Ornithoptera butterflies. So of those 284 pinned specimens that I ended up inventorying, 48 of those are actually type specimens that Treves donated to us. Which include types of varieties, subspecies, and color morphs, a lot of things that were very important in the collector trade and now are often synonymized under, accepted names. 99 of these specimens did not were collected before their listing date. 19 did not have their collecting dates listed, but we were able to find information in those letters and documents from Treves that told us when he actually owned those specimens. And then 19, I could not find any further information on their collecting dates or acquisition dates in the archive. Then with all these papered specimens, which is about half of the donation as well, I kind of went the opposite direction. So it was easier to see all the information that was written on the outside of these paper triangles and glassine envelopes. So overall, I had volunteers. Oh, and this whole project was made possible by all of our amazing volunteers. So they were organized into 3 categories, which was before June 1st, 1975, after, and then no date. And then for everything that was collected after and didn't have a date, then we identified every single one to see, or at least looked at them to see if they were a CITES listed species or not. The papered material has a lot of amazing data, and actually most of this material was collected by Treves and his friends and family. And I wish I had more pictures, but they were in these amazing like tea boxes and things from all over the world. And I wish I'd gotten to keep some, but all of these old boxes and drawers that we received the material in were saturated with naphthalene. So when we ended up acquisitioning the collection, we did throw those away, unfortunately. So yeah, basically I did all this work to see if all of these things were collected legally and did all these huge inventories and then proposed it to our curators at the time. And I did all this work for our former senior curator of entomology, Dr. Frank Thorsten Krell, and then the rest of the zoology department and with our registrar and everything. And they were like, sweet, we can accession pretty much all of the specimens. And our donation agreement as well also covered a line saying that the collector took responsibility for any specimens that weren't collected in adherence with those international regulations. So then we began the processing. So again, 122 drawers, 45 Schmidt boxes, pin material. Most of this was actually identified, but with outdated taxonomy and all of that very specific taxonomic that's like a form, of a color form, of a blah, blah, blah. So I had to kind of figure out what the updated taxonomy of that was. And I handed these drawers off to volunteers to database and rehouse. So I had like a rolling Word document. So every drawer, it had an inventory number. And then in each drawer, I basically listed from top to bottom in all the rows what all the specimens were and what they had been re-identified to. I printed all the labels and then put them in a plastic bag, slapped it on top of the drawer and handed it to a volunteer with a written out workflow. And then they rehoused and database and applied accession labels like that for me. And there's Kimberly there working away, one of my most amazing volunteers. Yeah, and so we basically took us a year to finish processing the pinned collection minus the types. So we finished that in December 2024. And a lot of Treves's material was curated by him in these pinning bottom drawers. It wasn't foam, but it was like a really gross old like cardboard, but we wanted to keep that curation that he had given to those because he had like additional labels often within the drawer that would apply to multiple specimens. So for these drawers that were already curated by him, we just moved everything over in the exact same orientation into our new drawers to keep that information and kind of keep the hand of the collector present within the donation. Some interesting things that are present in the pin collection. Who here has heard of this guy? Eugene Lemeul? Okay, yeah, so he was a giant butterfly collector and grew up in the French Guianan prison colony and basically was the largest or the only butterfly exporter in the country in the first 20 years of the 1900s. He described hundreds of new species, subspecies, forms, and varieties, released these really beautiful books. People bought the books and then, oh, guess what? You could buy the butterflies that were within the books there. So Treves did a lot of purchasing of these specimens and trading with other collectors. So a lot of the specimens from that pinned collection that Treves bought were from the Lemeul collection, absolutely fantastic picture there of him. So some standouts from the collection are Queen Alexandra male and female pair, which were amazingly photographed by our photographer and staff, Rick Wicker. So here's the male and here's the female, absolutely massive butterfly, terrifying to work with. I hate moving them. And then this really amazing bilateral gynandromorph of Morphodidius. So this guy or this specimen is exactly down the middle, male and female divided. So male is the more colorful blue on the right and female is the more black around the edge of the wings. And basically right away, I was trying to tell everyone in the museum that we work on this project because I was spending so much time on it. And I should mention that the Denver Museum of Nature and Science is not attached to a university and is very focused on public outreach in our exhibitions. So I was like, guys, we need to promote this awesome work that's happening in the collection that me and my volunteers are working on. So we got an article published in our museum magazine about the Treves collection, and Treves was interviewed by one of our staff members, which was really awesome. And there's Rick Wicker taking shots of the female Queen Alexandra's birdwing there. And then our former curator, Frank, he really wanted to get a case of these butterflies on display immediately. And he was the curator of this upcoming exhibit, Animals of the Rainforest. And so we worked with the exhibits team to design this case that would have the butterflies displayed. And so these were really fun days. So his three daughters, Victoria, Rebecca, and Francisca, came to the workshop with our exhibits team, and we artfully arranged the butterflies on these panels in three groups, which were the birdwings, which is Troides, Trogonoptera, and Ornithoptera genera, then Morphos, and then Papilio specimens, and created this amazing case that was then put in this exhibit. So there's me placing the specimens on this board. It was absolutely terrifying to do that. Don't recommend. And we actually did have the Queen Alexandra's pair on display for 3 months in this exhibit, which was really cool and special. And Treves then came with his family and got to visit the case and see it in the collection. He was regaling visitors about tales of them collecting all these specimens. And so it was really, really special to have him come in and view the specimens. And now this case is actually on permanent display in the museum. So the insect nook on the first floor of our museum, which has some really, really amazing interpretation direct from the 70s. So it's kind of at some different stages of being updated. But I got to be on the team for that and provide scientific knowledge on insects. And then we also inserted the case again in this exhibit. And so it's permanent-ish. So hopefully we check up on it every year, because we technically have a loan with the exhibits department that we'll need to renew every year, because I don't want those to get lost to just sitting on the floor forever. Make sure they're not getting eaten by pests or anything. And but yeah, feel really good about it. There's no UV light in this space. And the male and female Queen Alexandra's pair was removed for this longer installation of this case. Next steps for this collection is I need to finish processing the types. Got really busy, but there are some subspecies paratypes and stuff in there that are really interesting. And so we'll get those processed and uploaded. And I should say that pretty much all of the pin specimens besides the types are available on Ecdysis, and a lot of them are photographed, ventral and dorsal sides. So you're welcome to take a look. Let me know if you want to figure out how to search for them on there. We also got a lot of amazing books donated to us with this collection as well, including Zeitz's Macrolepidoptera of the World in French, and those Le Moule books as well that came with the collection. So those should probably be given to our archives department. And then we also need to process the papered specimens, which again is half of this whole accession. So 5,000 total specimens, half is pinned and half is papered. And these are more, not the Macrolepidoptera species. So it's got a lot more diversity in it. And a lot of them have really, really good collecting data. So we'll need to rehouse them, database them. I have many ideas. So we need to do that. And I'm excited to do it. Yeah, thank you to everyone, especially Treves. It was amazing getting to have him and his family and kids and grandkids in the collection, to see their collection in the museum. And thank you. Questions? Yeah. Mm-hmm. Yeah, so the donation of this person online wants to know more about the treatment of the donated material. Yeah, I had really amazing, capable volunteers handling and rehousing everything. We froze everything for 2 weeks before coming into the museum. Any more specifics? Yeah, we were very careful. Things break, but not that many. So yeah. Yeah. Anyone else?
We have in your envelopes, or are you planning on rehydrating and spreading all the things.
So this person is asking if we're planning on spreading the papered material or just rehousing them in clear envelopes. I would love to hear arguments to either side. Our former curator wanted to pin and spread them. I think we can rehouse them in clear envelopes and like database them, print out a card that has the collecting information on it, and then stick the specimen in front of a new envelope in the glassine. And then if a researcher wants, to spread them on loan or request that we spread some, like we can do that. But we're only going to have less and less space as time goes on. Yeah. One more. Yeah, so the commenter online was wondering about how we protected ourselves from naphthalene. Thank goodness these drawers like off-gassed somewhat quickly in the collection, but we did wear gloves when handling specimens. And I did wear like a mask, not a respirator, but just like a KN95 when handling them. And I encourage volunteers to do that as well. You can also just kind of feel it on your face over time. Does anyone know what I mean? So that wasn't ideal. But yeah, so we then threw away the old material that it was housed in the drawers and boxes, which people are like, you're throwing away the drawers. I'm like, girl, you do not want those. But yeah, so I trained everyone on safety on the naphthalene, and then we just got rid of the material as quick as possible. But now that smell isn't even present in our collection any longer, which was great. Yeah. I know, they were also falling apart. I know. Next time we'll talk. All right, thank you.
And up next, Life in the Fast Lane, Ligia Benavides Silva.
Thank you. Good morning, everybody. My name is Ligia Benavides, and I'm the curatorial assistant of the invertebrate collection at the Museum of Comparative Zoology. Today, I would like to give you a glance of a couple of initiatives that we have been developing at the MCC in order to preserve genomes and morphology of the type species housed at the MCC. And I'm going to give you a couple of examples of these initiatives in a group which I'm an expert, which is arachnids. So hopefully you enjoy it. The MCC is a repository of the four collections in the MCC are repositories for invertebrates. We have the entomology collection, which with more than 7 million of specimens and 11,000 type specimens. We have the invertebrate paleontology collection, which houses fossils of everything except arachnids and myriapods. And it contains about 1 million of specimens with around 10,000 type specimens. We have the malacology collection, which houses all the shells, and it contains about 500,000 lots and 5,000 type specimens, if I'm correct. And there's the collection that I'm part of, which is the invertebrate zoology. Up to now, it has about 1 million specimens and around 10,000 type specimens. In IC, we have both wet and dry specimens. The dry specimens, as you can see in the slide, we have the corals, we have echinoderms, we have ophurans. But the vast majority of what we have in the collection is stored in ethanol. Can be ethanol at 75 or 95%. 75 is most of the historical specimens, so from all expeditions, all donations. But from the 2000 onwards, we've been trying to store our specimens in 95% ethanol. And we try to keep them at minus 20 Celsius, minus 80, or in the cryo collection if possible. And why we do this? Because as you may know, the DNA integrity and yield is affected by the type of preservation, right? So things that are stored in 70% ethanol at room temperature, the DNA tends to degrade faster. And we want to preserve this resource. So with this in mind, we know the natural history collections are exceptional repositories of taxonomic knowledge and genetic resources. They provide historical and ecological data. And with this type of data, we can enhance our understanding of a species distribution over time. We also can study morphological adaptation to climate change. And with this, also inform the assessment of a species conservation status. Well, natural history collection and museums are excellent settings for research, right? We've seen that in the previous talks. However, there are a couple of questions that came up. And these questions are that how can we update methods for preservation of both phenotypes and genetic resources, and with this help facilitate research and serve better the community of scientists? And also, are the museums really sampling to preserve the diversity? With these questions in mind, a grant, our 3 PIs, Breda, Gonzalo Girivet, and Scott Edwards applied for a grant, the CSBR, which is the Collections in Support of Biological Research. They wanted to preserve the genomes of the type specimens in the Museum of Comparative Zoology, not just invertebrate zoology, but all the collections except insects, I believe. So the idea of this grant, the goal of this grant was to subsample and preserve in the cryo collection tissues of most of the primary types for a long-term cryogenic preservation. With this, of course, we wanted to preserve the DNA for genomic sequencing and provide the genetic material to the scientific community, right? So right now, we are storing part of the tissues in the cryo collection, and as I'm going to show you in a couple of slides. But in the future, the goal of the NCC is instead of sending the specimen of part of the specimen for sequencing, what we want is to in-house generate the genome or at least make the extractions and provide an aliquot of these extractions for the researcher, right? And with this, of course, we minimize the manipulation of the specimen and prevent that they get lost in transit. And I'm going to show you, actually, this one goes first. So the specimens are stored in our cryo bags at about minus 167 degrees Celsius, which is the gold standard for tissue preservation. So the idea of this is to have the type specimens and genome associated with it, and also morphological information. So of course, this work is been done by several people. Amongst them, we have Nina Black and Zoe Flores, and they have sampled around 8,000 specimens. As you can see in the slide, the tissue is placed in a cryovial with a barcode, and this is stored in a box that goes to the cryovac. Right. No. Oh. So these are the numbers. Sorry, I cannot see. So it's the top sample classes so far. We have, of course, arachnids with about 4,000 specimens in total, and we have subsample about 3,500, which is 81%. The remaining 20% we haven't subsampled because they are lost, or they are dry, or they have been preserved in formalin, which is not good for keeping the DNA. We have also Malacostraca, Diplopoda, Ophiurids, and some other classes. Well, I'm going to talk shortly about the phylogenomic approaches in systematic studies and how museum specimens come into play in this. So some of the phylogenomic approaches for systematic studies include the generation of genomes, transcriptomics, rat-seq sequencing, and techniques called target enrichment, which we have among them the ultra-conserved elements, which you might have heard about them, and the anchor enrichment sequencing. The disadvantage of some of these techniques, like the genomes and the transcriptomics, is that you need fresh material, right? And as we know, a lot of the specimens in the museums have been collected like more than 100 years ago. So that's where the reduced representation libraries, which are the UCs and anchor enrichment sequencing, come into play. With these techniques, we can generate genome scale data, which are relatively cheaper and more efficient, both in terms of lab work and bioinformatics, than generating genomes. We can resolve both deep and shallow relationships. And the advantage in the case of the UCs, the ultra-conserved elements, is that we can work with old and degraded samples, such as the things that we find in the museums. Really shortly, what are ultra-conserved elements? They are really short, about 200 base pair sequences in the genomes of all species that are conserved across different species. And why are they useful? And they are useful in both deep and recent because we have the core sequence, which is conserved. But if we move away from that core sequences, the variation increases. So that's useful for resolving population level questions that the researcher may have. So the development of the UCs together with the different milestones in DNA sequencing, new technologies, have helped to bring the museum specimens back to play, right? So we know that natural history collections play a critical role in biodiversity research. Museum specimens are being included more frequently on genetic studies. Studies show that pin and formalin-fixed specimens have been successfully sequenced. But up until recently, no tests on ethanol fixed specimens have been made, and ethanol in 70%. So this is why, with my two colleagues, Shahander Karavetian and Gonzalo Girivet, we asked the question of that can historical museum specimens preserved in ethanol at 70% be successfully used for modern phylogenetic studies? And we tested with opiliones, the daddy long legs. We extracted DNA of specimens from the collection, from the shells, and we successfully obtained sequences for specimens as old as 120 years. The tree that you see there, the phylogeny, shows in blue the specimens that were collected, that museum specimen, historical specimens. And as you can see, you cannot see, but it shows that the specimens were placed in the right clade within the opiliones. So that shows the efficiency of this method. Well, in addition of the genomic data, we are also trying to preserve the morphological information, right? How are we doing this? We are doing micro-CT and confocal microscopy of the type specimens. Here in that picture, I'm showing you the different ways that we mount the specimens in the MCC. Some of the specimens, of course, you don't want to dry the types, so we put them in a small plastic vial with ethanol and scan them. And some structures, like the pulps of some spiders, you can mount them dry, and that allows more high quality scan. We have two CT scans at the museum that we can use for this purpose. Let's see this place. Okay. So here are two examples of the things that I've scanned so far. Not most of the types. I'm scanning the types. One of the scanners allows really high resolution, so you can see morphology and some of the morphological characters that you need to identify the specimens, right? In addition of the CT scanning, we're also using confocal microscopy, in this case to document the morphology of the genitalic characters as well as somatic characters. The thing in green is the genitalia of Cyphophthalmi, which is a type of daddy long leg, and the other illustration there is that of a pseudoscorpion, illustrating some of the characters necessary for taxonomic identification. So ideally, or what we're working on is to deposit, make a morphosource, a repository of this morphological data for everybody to be able to look for the specimen that they are interested at and access the CT scan and the other morphological data. So you have a video that shows rotate the specimen as well as images. And all that information is linked on the MCC base. If you go to the MCC base and you click, you will be taken to Morphosource and you will be able to access the videos. And that's it. Thank you very much. Any questions? Yeah.
I have only slightly silly question, which is the Pilionids famously long, very skinny legs. Can you just take a leg off and get DNA out of that?
Yes. That's what we do. Sorry. Yeah. The person is asking if we can take a leg of the daddy long leg and obtain DNA. Yes, depends on the size. The bigger, of course, the better, but we've been successfully extract DNA of really small segments of the legs. Right? Yes. Okay. Thank you.
Hey, and up next, Natalie Dale-Skey with looking for the tooth fairy.
Hi, I'm Natalie. I'm curator of calcidoids at the Natural History Museum in London. And I'm going to be talking about a few things we discovered about our Mymaridae slide collection in connection with a digitization project. So Mymaridae, I'm missing a slide. Okay, never mind. I'm missing a slide. Mymarids are known as fairy flights, which is the excuse for the awful pun in my title. And even though you cannot see the image because I'm missing a slide, they contain the smallest known insect, Kikikihuna, at about 0.16 millimeter, and the reduced male of Dicopomorpha, a species name I can pronounce, at about 0.13 millimeter. So like most microhymenoptera, the small size makes them very challenging to collect, preserve, let alone study. And generally speaking, there's very few people working on them, and we know who they are. They are professional entomologists. So our Mymaridae slide collection was included in a big digitization effort along with a few other calcidoid families, which started in 2015. In fact, I talked about the infancy of this project at the first ever ECN conference I attended. And the project took place in two phases. first teams of volunteers scanned slides in bulk and created stub records in our database. And in the second phase, labels were transcribed on the crowdsourcing platform Notes for Nature. And there was a much longer unofficial phase consisting of me going through the data, amending it, and in a lot of cases also putting it in a format compatible with our database. So in the first phase, volunteers scanned slides in Treves, 100 slides, uploaded it to the software in Select, which you've already heard about. The images were segmented automatically and tagged with the QR codes and taxonomy. And the resulting images were used to create stub records in the database. So at the end of phase one, what do we know about the part of the collection we digitized? Not that much. Basically, we know how many we have of each, here summarized by family. But more importantly, we have all these images. So in the second part of the project, all the images were uploaded to Notes for Nature, where volunteers all over the world could help with transcribing the labels data. And these are, excuse me, the items we asked for: date and country of collection, collector, host insect and host plant, whether the specimen was a type, and the BM registration number. And as part of this phase, the person who was in charge of the volunteer program at the museum at the time organized visiteering days. So visiteering is a really nice concept where people who don't have time to commit to medium or long-term volunteering could come to the museum as part of a group for just one day and work on a specific project. So in this case, we had a room full of computers, and they would spend the day transcribing a Note for Nature. But of course, I would give them an introduction to chalcidoids, to scientific slides, and I was there during the day so they could ask questions if they had any. And as part of this visiteering, people started asking about slides like the one you see here on the right. And they were asking because you'll see the labels are in two different orientations. So unlike standard slides where labels are either landscape or portrait, these ones, because they're in two orientation, it's highly unusual and also very annoying because it requires an additional rotation to read the labels. And that got me thinking, it's true, we have a lot of those slides and I don't actually know where they are from. All I have is these initials, SB. So I did what I always do when I have questions about a collection. I asked John Noyes, who already second picture of John this morning, chalcidoid expert at the museum who has a phenomenal knowledge of our collection. And he said, oh yeah, that's Scarsdale Brown. He was a dentist in Bournemouth, that's in the south of England. And we were surprised he gave us a big Mymaridae collection and no one knew who he was. So now that I had a name, I did a bit of searching and I did not find much, but I found this note published by Brown in 1972, where he describes how he was able to collect live Mymarids from the sticky leaves of a tobacco plant. And also he was mentioned in a report from the Bournemouth Natural Science Society, which taught us that he was primarily a microlepidopterist who had diversified into Hymenoptera. So armed with this knowledge, we published a blog on the Notes for Nature platform and moved on with the project. So by the end of the project, we now have a lot more information on the slides that were digitized. You see here for the items we had asked for how many slides had the information. You can see it varies between the different items. And some of it is because not all information is on all slides, but some of it is also because some type of information was much more difficult for volunteers to find on the slides. The worst was the host insect and host plants. Host insect, the most frequent issue was people would put the name of the specimen as host insect. And we also had some oddities like this one that Sergei Trapitsin, who is here, but I don't know if he's in the room, who identified the specimen was listed as the host insect, which amuses me no end to this day. And this one, a brown slide under the host plant field, we had this comment, your interface is making this task near impossible. And I can't really disagree. I think we were asking possibly for too much. But still, we have a lot of information. We know when those slides were collected. And for now, I just would like you to keep in mind the big peak in the early 80s. We know where they were collected. That's the sort of spread you'd expect with a few countries with a lot of specimens and a lot of countries with very few specimens. If we focus on the left of the graph, you can see what it's mainly collected in the UK, and you can see also comparing corrected data. So once I have gone through the data with original data, that the quality is pretty good. The main difference in a lot of cases, United Kingdom was amended to England, but that's just because this option was not available on the website. And of course, we know who collected the slides, and SB reappears. But you can also spot immediately a very familiar problem, which is that one person appears under different versions of their name. So focusing on SB, that's all the different ways he appeared in the data for a total of 444 slides. And in fact, once I had gone through the data, adding the slides where he had been missed, I saw that 690 of the slides digitized were collected by Brown, that's 11%. Looking at just Mymaridae, 30% of the slides were collected by him. And looking at just UK Mymaridae, almost half the slides digitized were collected by him, which to me was just staggering. Who is this guy? So first, what can our own collection tell us about him? So we know he was based in England, and this world heat map of these specimen localities confirms his focus was in the UK. If we zoom in on the UK, we can see his focus was actually very much where he lived in Bournemouth and to a certain extent the surrounding area. If we looked at how and when he collected, if you remember the peaks in the early 80s from the general data, I think we have found a culprit. So these two years, he collected with a malaise trap. The years before, specimens were swept or bred, which you could think possibly was a sort of in association with looking for microlapse, but clearly a malaise trap must have been put up, if not specifically for microlapse, at least for non-Lepidoptera. And we don't know why, but it might not be a coincidence that Bob George, another British entomologist who worked on Mymaridae, moved to Bournemouth in the early 80s and started a company supplying malaise traps. So maybe he told Brown, hey, why don't you try one of those? And that's what happened. So if I step outside my collection, looking just at digitized specimens, I found 145 LEPs that were collected by Brown. It confirms the geographical focus in the south of England, but with a different range of years. Here, the earliest specimen is in 1919. So one thing we know, considering our latest specimen is in 1985, the guy was a keen collector. That's 66 years of collecting insects. I also found a few Hemiptera, and in the pin Mymaridae, an additional 75 pin Mymaridae with the same range of years as the slides. And if I'm very brave and steps into the real world, what do we know about Brown? So his full name was Sidney Charles Scarsdale Brown. He was born in 1903, lived to be 100. We know he was a dentist. He spent most of his life in Bournemouth and was a very active member of the Bournemouth Natural History Society. He was married to Margaret Brooks, a famous British lepidopterist who published those two books and was, I believe, a pioneer in using photography of live insects. And I also found this note telling us that when Brown was around 90, he decided to leave his beloved Bournemouth and move to warmer climes. We don't know where he went, but he didn't like it that much because we know he did die in Bournemouth after all. So looking at his publication, he did publish a few papers, mainly on LEPS and history of entomology, but more strikingly, he published a lot of records, not just of insects, and also an incredible number of notes and natural history observation, including here my favorite about a sparrow beating up a moth against a branch to kill it. And he's also very frequently mentioned in other people's notes as having helped them with collecting, showed them something. So the general impression you get is of someone who was not only very knowledgeable about the natural history of his area, but also very keen on sharing. So now we know what his slides look like, we know what his spin specimens look like, and we know what his publications look like. But what did he look like? Well, here he is. Now, I don't know about you, but this is exactly what I want my dentist to look like. This guy is not going to hurt you. So do we have any further evidence that Brown was just generally a very nice guy? Yes, we do. You might remember the blog that had been published. Years after the project had concluded, we got this comment from someone who had been a patient of Brown when they were a kid, and telling how Brown had nurtured their interest in entomology and crediting him with their continued, not only theirs, but also their son's interest in natural history, concluding Mr. Scarsdale Brown cast a long shadow. So with this, I want to thank Jeff Turnock and Janet Davis at the Dorset Museum, who were very helpful and generous in sharing information about Brown and his collection. Mark Sterling in London, who told me that even though Brown was an unknown among hymenopterists, he was well known as a collector amongst lepidopterists. John Noyes, without whom I would never have known who SB was, and of course all the people who worked on the digitization project. Thank you. And I believe I have no time for questions.
All right. All right, and up next, Shelby Grice with who will tell us about a spectacular scale collection.
Hello. My name is Shelby Grice. I'm actually the outreach coordinator for the Mississippi Entomological Museum in Starkville, Mississippi. And I'm going to preface this with the fact that I know little to nothing about scale insects. So forgive me in that this is just going, this is a really cool collection. This is a really cool lady. I don't know anything about it, but maybe some of you do, and some of you are interested in coming to see. But let's get started. So, who was Mary Gladys Hoke Lobdell? She was born Mary Gladys Hoke, May 13th, 1895, somewhere in Panola County, Mississippi, which you don't have to know where that is the middle of nowhere, Mississippi. And of course, as to be expected with somebody whose career was primarily in the 1920s, a little bit before that, a little bit after that, there's not a whole lot of information about her that you can find by a simple Google search. So, what do we know? The first thing is underneath her beautiful portrait right there, what was written on her gravestone: scientist, teacher, wife, mother. And I do think that that is a pretty fantastic summary, and I hope that somebody thinks of me that lovingly for my headstone as well. So she was from 1895 to 1981, a scientist for about as long as I can kind of figure out. Her family seemed to live within the natural world and had a fascination with it in some way, shape, or form. So have some fancy correspondence from her, some very old letters. Miraculously, the Mississippi Entomological Museum has a lot of letters from her and from other people sending things to her. So through some of this, we know that she graduated from the Mississippi University for Women with her bachelor's degree. Back then, it was the Mississippi State College for Women. And then she got her master's degree from the University of Illinois. She's published at least a dozen articles. Again, it's kind of hard to find things, especially when she gets married and then her last name changes. So then you've got the added thing of, oh, is she going by Hoke? Is she going by Lobdell? Is she going by Hoke Lobdell? I'm sure that there's room for error in my estimates up here. And something that I found particularly interesting, I'm doing graduate research on grasshoppers, and I'm having to get pretty heavy into DNA. And when I looked at her thesis from the University of Illinois, she wrote on the mouth parts of Plecoptera. And you know, I had to sit back and go, I wish that I could just look at the mouth of something and be able to publish that for a thesis, rather than kind of dive into people telling me what the DNA says and me trusting them and then like interpreting it later. So this up here, and I'm sure that it's going to be hard to actually read, but it looks really cool. And it is one of the originals for her offer letter to be a professor at Mississippi State University, back then the Mississippi Agricultural and Mechanical College. And this is for my folks with Mississippi State, we have the Harned Biology Building, and this letter was from Dr. Harned to Gladys, offering her a job working in entomology and teaching for the Department of Zoology and Entomology, which I wish was still a department today. But they offered her $75 a month to be a professor, and she took the job. So that was in 1918, and that is really when you start to see her publications come out. Her thesis was actually published in 1921, so took her a little bit after graduating, but no shame in that. We all have to go at our own pace. And something else that's very interesting is, of course, she's described her own scale insects. And in a lot of these, so I have no idea what I'm looking at. That is a blob with legs, and it's very cool, and it's a beautiful illustration. But this is one that she described. And forgive me for my pronunciation, Phrysia clavicetta. Very fancy. But not only did she describe these and go into all of this detail, her sister was actually a scientific illustrator. Her name was Sarah Hoke. Sarah Hoke. Oh my goodness, it starts with a D. Anyway, we'll get to her. DeBoard. There it is. It came to me. So during her time at the Entomology Museum, Gladys, of course, it was not as it is today. And today we are fairly independently funded. We do our own grants, we work through our own things, but at the time it was run by the Mississippi Plant Board. And so it was super particular about what they focused on. Of course, I mean, we still focus on plant pathology today, but it was very limited scope in what can we do for the agriculture of Mississippi. And in 1889, Leon Hepner, one of our old predecessors, one of the first directors of the museum, the associate curator at the time, he had stated that the collection had over 575,000 specimens. And today we have over 3 million. And I think that's just a fun little comparison. But this is Gladys's scale collection. So we still have this handmade set of drawers, and nearly all of them are filled with slides in paper. And it's a very interesting mechanism. It's in several pieces. And you can see here, you kind of have to open one drawer in order to open others. One will lock the others, but once you've opened that, then there's more that you can see. And it's actually in 3 separate pieces. You've got the legs of it, and then you've got 2 different sets of very heavy bits of lots and lots of delicate glass. Ask me how I know. We had to move it. So her personal collection here contains over 4,000 individual slides, which, oh my gosh, that is a lot of really focusing in on getting a teeny tiny bug onto a slide for you to be able to see, and still preserved. It's fantastic. She's got lots of different, both native and ones that are exotic from other countries and things like that. And most of the drawers are filled to the brim, just like this. And then she's got a very particular types area, and they're all to themselves. And that's almost 100 slides right there. Now, I was not brave enough to go in and take out any of her slides because God forbid I move things around and it gets shuffled and nobody knows how to put it back together. So I don't have pictures of her slides, but something that our curator was able to find for us was these boxes of identified scale insects. And of course, we were like, hey, that's really exciting. We've got more things to add. We're finding these things. And then we were a little bit hesitant when we opened it up. You're like, oh, well, I see a little bit of damage at the top there. Hopefully it's not too bad. And then you empty the box. So, needless to say, a lot of it was unsalvageable. You can't bring it back, but we can document maybe what it was. Unsure what it is in the moment, at least. And though there were some that we couldn't get any information out of, there were some things that were pretty interesting. This, I believe, is a letter from her father sending her some leaves. Now, I didn't see any correspondence in it that really explained what it was. Again, I'm only so brave. I'm not going to really pick apart an envelope that's from the 1800s. But you can see a teeny tiny white speck on the top of one of those leaves. And I think that that's a scale. And that's probably what he was sending this to her for, is like, hey, look at this cool thing that I found. I wonder if you know what it is. And then something that I'm wondering what in the world it is, we found this really cute round cardboard-ish container filled with that. I don't know. Maybe it's galls. Maybe it's the mites. I have no idea. Or the scales rather, not the mites. But it's very interesting. And perhaps that there is something in there that explains what it is and it has an association. But there's a lot in those boxes. And again, my bravery only goes so far in digging around old documents when I'm not trained for this. I don't want to mess anything up for the future. So this, of course, stumped all of us, and hopefully one day we can bring these associations back together and really be able to understand these pieces of this research that she did. I mean, this woman obviously cared a whole lot about her work. She has so much invested into it. Decades of research, of publications, not only identifying, but also going out and just documenting what's around and saying, hey, these are the things that I found in Mississippi. She later moved to North Carolina and did the same thing there. So, who knows what we can find out from her collection? I sure don't. Now, she contributed a lot more than just what I've covered, because of course, there's a lot we don't know about her. And I mean, even just her being her contributed so much to the scientific community. I mean, in my mind, she might as well have been the reason that I'm here. She was the first ever female faculty member at Mississippi State University. I mean, that's insane. That's amazing. And it's so wonderful to be able to put a name to that legacy, right? So for all scientists, not just entomologists in Mississippi, for all scientists in Mississippi, especially if you're female, that's amazing. And we have her to thank for it at a large extent. Now, I did mention that her sister was involved in some scientific illustrations, and here is some of it. So she was very talented. She did not only some of these illustrations, she did a lot of Microlepidoptera illustrations as well. So this is actually from another female scientist that I don't know a whole lot about, but now I'm going to do a deep dive later when I get home, is Miss Louise Russell. And she did her own species descriptions and had plenty more in the 1940s to do. So we've got from one stepping stone to another, there's more and more of these female scientists that we don't really know about. Now, this particular image is from Miss Russell's descriptions of 9 species of Aluroplatus from Eastern North America. So, very cool. Another thing for me to put a pin in later, and then maybe I'll convince other people to look into her. Who knows? This is Gladys. And I'm going to go ahead and say in her prime. This is the youngest image that I could find of her. Very interested in her microscope. Reminds me of myself when I finally get to do lab work rather than doing all my planning and coordinating and all that, which I love, but also I miss the scope. And on Gladys's scale collection, we do have her obituary. So it is very loving. It talks about, how much she contributed to science and how much she affected the others around her, being a wonderful teacher and, just her. We're thankful for her. And I love that we were able to keep this obituary with her collection to remember, she was more than just a scale insect researcher. She was more than just the scientist. She was a mother, a teacher, a wife. Very fun things. And just to wrap up, I wanted to thank everybody for giving me this opportunity. Again, I don't know anything about scale insects. I hope that somebody else is really interested in it and will come and visit me in Starkville at the Mississippi Entomological Museum. We have a lot of cool stuff. This is not our only hidden gem, and I hope that you will give some love to this fantastic historical collection. Thank you. Good. Okay, I have time for one question. Thank you very much.